Hi everyone, my name is Lauren Hill-Bowen from Florida International University in Miami. Thank you to the CPDD conference organizers for providing an opportunity to share my research, and thank you all for listening today. The title of this presentation is Convergent Network-Level Brain Alterations Across Drugs of Abuse, a meta-analysis of structural MRI studies. Drug addiction is associated with an array of alterations in emotional, cognitive, and reward processing, which maintain drug use and make targeted treatment difficult. Over the past 25 years, non-invasive brain imaging techniques, such as magnetic resonance imaging, has provided insight into the functional and structural brain changes linked with these altered mental operations. Prior work has identified reduced gray matter volume in specific brain regions that correlates with clinically relevant behaviors and potentially contributes to the development and or maintenance of addictive disorders. For example, studies on alcohol use disorder have demonstrated relationships between gray matter volume and self-reported impulsivity, such that reduced medial prefrontal gray matter correlates with higher impulsivity scores. Similarly, when considering methamphetamine dependence, a negative correlation between gray matter volume and behavioral performance on a go-no-go task, an objective measure of impulsivity has been documented such that reduced butamen volume is associated with lower performance. Importantly, such gray matter alterations also correlate with the amount of drug use, where, for example, more cigarettes smoked per day is linked with reduced structural integrity in the insula. Despite enhanced insight into structural alterations associated with addiction, neuroimaging studies are often limited by focusing on the impact of one specific substance and by focusing on individual brain regions. In contrast, neurobiological theories of addiction often emphasize brain network level impacts of substances across pharmacological classes of drugs. One possible way to bridge this gap between theory and practice involves the application of data-driven meta-analytic techniques. Such techniques allow for the synthesis of the primary literature to provide consensus views. The objectives of this study were to utilize coordinate-based meta-analytic techniques to characterize the functional profiles of structurally altered gray matter regions consistently observed across substances of abuse, and to utilize data-driven techniques to delineate what the potential behavioral implications of such brain alterations may be. We sought to achieve these objectives with three primary aims. First, focusing on aim 1, we aim to identify consistent regions of reduced gray matter volume in substance-using individuals compared to non-using controls across 5 pharmacological classes of drugs: alcohol, nicotine, opiates, cannabis, and stimulants. We identified a total of 87 studies and performed a meta-analysis using the activation likelihood estimation algorithm, a voxel-based method for determining statistically significant spatial concordance within a set of studies. All 87 studies across the 5 drug classes were included in the analysis to identify regions that consistently demonstrated reduced gray matter volume. Convergent gray matter reductions were observed notably in the cingulate gyrus, medial frontal gyri, superior frontal gyrus, and multiple regions within the insula. Previous literature has implicated similar regions, including the ventromedial prefrontal cortex, insula, and anterior cingulate, as being critically involved with inhibition, motivation, and reward-guided behaviors. More specifically, the VMPFC is engaged in goal-directed behaviors, performance monitoring, and inhibition, as well as reward-based learning. The insula is thought to be critically engaged in the perception of the internal bodily state, with more posterior regions involved in homeostatic motor functions and more anterior regions involved in motivation. Further, the connectivity between the insula and anterior cingulate has shown to be essential for choosing and mobilizing adaptive behaviors, such as reward-guided choice. Second, we aim to delineate those brain regions with which these structurally altered areas functionally interact with, thus delineating an extended network of impact. Working under the assumption that this larger extended network can be decomposed into smaller subnetworks, we employed a principled clustering approach. We first evaluated task-free resting state and task-based meta-analytic functional connectivity assessments for each structurally impacted seed region. We then examined the conjunction of these two maps to generate a connectivity profile that was consistent across both brain states per seed. Next, we identified all regions in which at least 3 of these seed regions maps demonstrated overlap. We termed this larger network an extended network, and it represents a set of core regions strongly connected to multiple original structurally altered regions. Finally, hierarchical clustering was applied to the task-free and task-based connectivity maps of the extended network regions to establish subnetworks within this larger network. Regions functionally connected to those showing drug-related structural alterations notably included the anterior cingulate cortex, posterior cingulate, inferior frontal gyrus, insula, superior temporal gyrus, and putamen. This map is what we termed an extended network of impact. Hierarchical clustering of each region's functional connectivity profiles identified 3 distinct cliques or subnetworks. Regions making up subnetwork 1 in red demonstrated a functional connectivity profile with the posterior cingulate, subgenual anterior cingulate, medial frontal, parietal, and temporal regions. Subnetwork 2 in green demonstrated a connectivity profile consisting of the putamen, inferior frontal gyrus, and dorsal anterior cingulate. Finally, subnetwork 3's profile included inferior and middle frontal gyri, insula, cingulate, and parietal regions. This apparent shift from ventral ACC to dorsal ACC to posterior supplementary motor area seen from subnetwork 1 to subnetwork 3 potentially is consistent with a hierarchy of information flow seen in spiraling frontal striatal circuits. Finally, we aim to characterize the putative mental operations linked with each subnetwork via functional decoding. This approach utilized a large neuroimaging results repository called the Neurosynth database. We first calculated an average task-based connectivity map between regions within each subnetwork. We then correlated each subnetwork's averaged map with maps in Neurosynth that correspond to each term. This allowed for the identification of top functional and anatomical terms linked with each subnetwork. Results provided enhanced insight into the psychological processes associated with each subnetwork in a data-driven manner. Subnetwork, or clique 1, was composed of regions displaying similar topological properties as the canonical default mode network. Neurosynth decoding included functional terms of retrieval, engaged, mentalizing, and autobiographical. Results indicate that this clique was associated with internally oriented default mode processes. Subnetwork 2 included similar topological properties as the canonical salience network. Neurosynth decoding included functional terms of reward, incentive, anticipation, and motivation. Results indicate that this clique included features commonly implicated in reward-related paradigms and salience attribution. Finally, subnetwork 3 was composed of regions with similar topological properties as the canonical central executive network. Neurosynth decoding included functional terms of task, demands, and conflict, and working memory. Results indicate that this clique was associated with executive functions across tasks requiring high cognitive demands. This study sought to utilize coordinate-based meta-analytic techniques to characterize the functional profiles of structurally altered gray matter regions consistently observed across substances of abuse, and to utilize data-driven techniques to delineate what the potential behavioral implications of such brain alterations may be. Through the application of data-driven meta-analytic techniques, we identified convergent regions of reduced gray matter volume across pharmacological classes of drugs in regions critical for interoception, motivation, and cognition. We subsequently characterized the functional connectivity of these regions to delineate a larger extended network representing a core set of regions strongly connected to structurally altered regions. The extended network was found to be composed of 3 subnetworks that closely correspond to the tripartite network parcellation, where the dynamic interactions between and within these large-scale brain networks has shown to have important implications in stages of the addiction cycle, such as withdrawal. Thank you all for being here today and listening in. I would like to acknowledge my lab at FIU and all those that have contributed to this work, with a special thank you to Dr. Michael Rydell. I will be available at the live Q&A session on Tuesday, June 23rd at 3:30 to discuss this work further. See you then.